Assalamu alaikum. In this video, we are going to talk about the effector function of the T cells in detail. There are two important effector functions of T cells. First is a delayed hypersensitivity and second is a cytotoxicity. First, we will talk about delayed hypersensitivity. The delayed hypersensitivity reactions are produced particularly against intracellular microorganisms, and these organisms include both the fungi such as histoplasma and bacteria such as mycobacterium. The cells of the delayed hypersensitivity include macrophages and CD4 cells, and particularly the Th1 subset of the CD4 cells. Important lymphokines that are produced in delayed hypersensitivity are the gamma interferon macrophage activation factor and macrophage migration inhibition factor. CD4 cells produce these interleukins or lymphokines, and the macrophages are the ultimate effector cells of the delayed hypersensitivity. Any defect in all these processes can cause infection. Now let's move toward the cytotoxicity. The cytotoxic response of T cells is concerned primarily with the virus infected cells, tumor cells, and the allografts. In response to the viral infected cells, the CD8 positive T cells are activated and they recognize the viral antigens on the surface of the cells along with the MHC1 protein. Free viruses that are present in the extracellular spaces are also presented to the helper T cells, and free viruses are presented to the helper T cell in association with the MHC class 2 protein, and the helper T cells are then activated. They produce interleukin 2, then the interleukin 2 activates the cytotoxic T cells, and then the cytotoxic T cells they migrate to the site of infection. They produce specialized proteins called as perforins. These perforins produce pores in the cell membrane of the viral infected cells. Through these pores, the inner content of the cell leaks out of the cell and the cell dies. After killing the virus infected cell, the cytotoxic T cell is not damaged itself and it can continue to kill other cells which are infected by the same virus. It is important to note that the cytotoxic T cells has no effect on the free viruses. It can only kill the virus infected cells. In addition to the direct killing of the virus infected cell by the cytotoxic T cells, there is also another mechanism through which the viral infected cell or virus infected cells can be killed, and this include antibodies and phagocytic cells. And this process is known as the antibody dependent cellular cytotoxicity. In this process, antibodies bind to the surface of virus infected cells, and macrophages are the cells that recognize those antibodies bound to the virus infected cells, and then those macrophages kill the virus infected cells. The antibodies that are involved in this process include the IgG for the virus infected cells, and if the disease is caused by the helminths or otherwise known as worms, the antibodies that are involved in the killing of these worms are the IgE. IgE binds to the surface of the helminths, and the eosinophils are the cells that recognize that IgE bound to the surface of the helminth. Then a protein that is present in the granules of the eosinophil called the major basic protein is released from the cell, and this protein damages the surface of the worm. Now let's talk about the tumor cells. Tumor cells, when they develop, they produce new antigens on their surface, and these antigens are recognized by the cytotoxic T cells or CD8 positive T cells, and these antigens are presented on the surface of the infected cells or the tumor cells along with the MHC class 1 protein, and the cytotoxic T cell recognizes them and kill those cells, and this process is called as the immune surveillance. In the case of allografts, the cytotoxic T cells recognize the proteins that are present on the surface of allograft as foreign antigens. The proteins such as the MHC class 1 protein, MHC class 2 protein that are present on the surface of the allograft, they are recognized as foreign by the cytotoxic T cells, and these allografts are then destroyed by the cytotoxic T cells.